Welcome to Biotechnology. Here we're talking about extraction of DNA from a cell. So we're going to take some human cells, namely mine. Those are up there on the left. The cell has a cell membrane. It has a nuclear membrane. Outside the nucleus there are DNases, which are going to try to try to break down DNA if they get in contact with it. Inside the nucleus, the DNA is wound around proteins that are specially there to organize it so it's easier to find, just like the wire on my microphone is wound around a little bit of cork so that I can not get it tied in knots quite so badly. The steps that we're going to go through are as follows. We're going to put the cells into a soap, SDS, sodium dodecyl sulfate, with some buffer in it. The buffer will keep the pH constant, which will keep the charge on the DNA constant, and will help the enzymes in the next step work properly. The SDS will break down the membrane because it is a soap, and it will integrate itself into the membrane and break it up. The membrane is only held together by hydrophobic interaction, so soap is obviously going to mess that up. That's why soap is so painful if you get it into your eye, because it breaks down the cell membranes and kills them. Then all of the inside of the cell will leak out, and we will give it a shake, and then we'll add some protease, which is an enzyme that breaks down proteins. That will kill the DNases, but it'll also break down the spools, because they're made of protein, and allow the DNA to open up into its normal form. In the third step, we're going to treat it with salt and cold and isopropyl alcohol, which will cause it to the DNA to clump together because it's not very soluble in non-polar solvents, and particularly if there's salt in there, because the salt allows it to stack together as plus minus, because DNA has a very large negative charge. On we go into the lab. Welcome to Biomaterials, Biotechnology, in which we're going to extract some DNA from me today in an experiment that we can purchase from Biorad, but it's exactly the same way that you would normally extract DNA from cells. So what we're going to do is I'm going to first brush off some cheek cells inside my mouth. So it'll take a while, maybe fast forward over this bit. Well, so now, without swallowing, I'm going to take this water, it's normal tap water, and I'm going to squirt it around my mouth and spit it back into the tube. That's kind of gross. So what have I done? I've knocked some cells off the inside of my cheeks, and I've rinsed them out with tap water. That's why I'm not wearing a lab coat today, because obviously it would be kind of stupid if I'm going to put water in my mouth and I'm wearing a lab coat. It doesn't really fit together. So we can look at the solution. It's kind of gross. It's got all kinds of bits of cells in it, spit obviously as well, and the water. Now we're going to add some what they call lysis buffer. The lysis buffer is buffer to stop the pH from getting out of hand, because obviously if we're killing cells and causing all kinds of damage, then the pH might go somewhere weird and cause more damage to the DNA that we want to save. And it's also going to contain some soap. And the soap is a standard dishwashing soap, effectively. It's pure science version, sodium dodecyl sulfate. And what that does is it enters into the cell wall, and the cell wall is made up of effectively of soap molecules that are all organized in a layer, bilayer, so with the water-liking part outwards and then fat-liking part inwards, and the soap will destroy that by entering into it and dissolving the fat effectively. So we're going to add that. I have to check how much, so I will do that now. So the instructions say to use 2 milliliters of lysis buffer. we will do approximately that. Normally we would be using a centrifuge tube, which is not so easy to see through. So this is the lysis buffer. It's purchased from them. It says on it, it's got TRIS, which is a buffer, and it's got SDS, pH 8. The pH will come in handy later because we're going to add an enzyme, so that will be important. I'm going to turn you to here. Zoom in a little bit. So that is our cells in the tube, and I'm going to add, oops, I'm looking in the wrong place, I'm going to add the lysis buffer, which is a TRIS buffer, and sodium dodecyl sulfate, which is a standard buffer solution. I'm going to take 2 milliliters of this, because that's what it says, 1, 2. That's having a slight effect. We have to stopper our tube like this. Obviously, if we had a centrifuge tube, it would be easier to do that, but you wouldn't be able to see as well. They're not as clear, which is why I've got this. Now you can see there's already things happening. Ooh, it's kind of gunky already. It's already changing and getting more thick, and particles are appearing in there that weren't there before. Can you see that? There. It's getting all gloopy already. That's because the insides of the cell are coming out, and there are a lot more polymers and all kinds of things in there. We need to get a move on now, however. We need to add a protease, which is an enzyme that's going to break down the DNA ases that are now free because we've opened the cells up. So we're going to do that now, so that we don't get, so we don't expose ourselves too long to it. That's in this container here. Whoops. Focus. That's in this container here. It's just a protease plus a lot of salt. The salt is there to help a later step. Let's pop that in there. Maybe all of it. Whoops. Why not? Okay, let's give it another couple of twists. This shouldn't really do anything immediately. The enzyme shouldn't really do anything noticeable because it's breaking down a protein. Whoops, let's go on here. It's breaking down a protein that is there anyway. So it's not really changing the consistency. It's all of the other polymers that are going to do that. Let us go and put that in a water bath. So I'm back. I've got my water bath. Let's go down to it. That's my water bath. It's a very simple water bath, just a container with water in. It's at 50, wow. So I need to drop this in there, and that will be in there for 10 minutes. This is going to take a long time, so we don't need to watch this. We can watch it in fast forward. So I'm going to switch to time lapse, which is easy. Okay, so we've had our 10 minutes. I probably will insert a little bit of instruction in between, a little bit of explanation. We can see that our liquid is still thick like it was before. So kind of weird, it flows not quite like, flows not quite like water should. I get up closer and focus. There you can see it doesn't flow like water does. Depth of focus isn't great. There we go. So what I'm going to do now, let's keep it in focus, is move it into some ice water to cool it back down again. And then we're going to try to crash out the DNA. We're going to do that with a combination of the salt, because the salt will neutralize the charge effectively. The sodium ions will sit next to the anions on the DNA. The DNA has negative charge on every rung of the ladder effectively, so it has a very large negative charge, and we were not going to be able to pack negatively charged molecules next to each other. So what we need is some cations, some positive ions to go in between, which is why we've added a lot of salt. Then we're going to add something that is going to change the water to make it less polar, to make it less watery, and that it will be an alcohol. It has to be able to mix with the water, so we're going to use isopropanol, and we're also going to use cold. So I'm going to do it in this ice water, or at least I'm going to cool it down first. So that's reasonably cool. I'm going to go and get some pre-cooled propanol, isopropanol, which mixes with water, and we're going to fill up the tube with that carefully, balancing it on the top, and then we will observe it, hopefully separating the DNA. What we can see in our tube is that it's got even more viscous than before, because the DNA has now been freed from where it used to be. The DNA, in normal case, is wrapped around little protein molecules like a type of spool system, so that it doesn't get tangled around in the cell. It's much easier for the cell to organize it if it's sort of wrapped around little molecules, and that protease, a protein dissolving enzyme, will have broken down those little spools and freed the DNA up. It will have got rid of a lot of the protein from the solution, which would otherwise interact and cause issues and be chunky, and there'll be a problem. So now we've got soluble soup of short bits of amino acids, so short chains of amino acids, bits of protein, and the cell material and the soap, and some sugars as well. And the only proteins, the only polymers in there should be the DNA and some RNA. Okay, so our next job is to add some isopropanol. I've zoomed into the tube so that you can hopefully see what's going on. We will apply a bit more light and see what we can do. I'm going to add the isopropanol very carefully so that it floats on the top, which helps the whole process. So here it comes. What will happen is, as the isopropanol interacts with the water, it will generate a zone that is very non-polar, and the DNA will start to precipitate there, and then it will enable it to collect. So it's to help nucleation, to help the process to start, that we balance it like this. I'm going to leave it balanced for 5 minutes, which is what the instructions say, and I'll put another time lapse on so that we can watch it do it. I might turn some light onto the situation. Or so there we go. Now we can go back into time-lapse mode. So we can see that we've developed a layer. Let me see if I can bring it closer to you. Not sure how close this camera can focus yet. Pretty close. So we can see we've developed a layer here, a cloudy layer, then there's a clear layer, and then there's the stuff on the bottom. This is isopropanol on the top that's never mixed. So we're going to now invert this I'm going to zoom out for you so you can see. I'm going to invert this a few times, and you can see it forms clumps fairly instantly. I'm just motivating it to build clusters of DNA that then fall to the bottom. You can see it's all stringy and rather disgusting looking. And you can see how I'm holding my let's get that to focus there. Yeah, let's move it closer. So what we can see now is our DNA, or my DNA, that is on the bottom here as a clumpy, thick layer of polymer. It's obviously got a lot of water associated with it still, but most of the other gunk is in this liquid. It's still white, so it still contains particles of something, particles of proteins, particles of other materials. It's still a mixture, but the protein, the DNA, sorry, has clumped together and fallen to the bottom. If we leave it a long time or centrifuge it, then we can get it to all stick together. That's fallen. Got one to float to the top now. So that is our, oops, hang on, there. That's my DNA. Oh, sorry, I keep moving it towards me where I can see it, which is not where you can see it. Let's make the camera focus. So there it is. And if I turn it on its side, I can probably tumble it. There we go, that's the clump of DNA in there. Yeah, there we go, we can tumble it around in a horrible way. And that is just polymer, just like any other polymer that has been precipitated from a solvent. Okay, so maybe 2 hours have gone past. I got distracted by other things, so let's see what has happened here. It's warmed up to room temperature, obviously, but the DNA, see how close we can get. The DNA has stayed solid in there, and it shrunk a little bit. There we go. Oops, sorry. It shrunk a little bit into this sort of clump because it's lost a bit of water. So that's exactly what we would expect. The normal process now would be to take that DNA and do something with it, because now we can get rid of all the other material up here. We could wash it, and then we would have to do the next process, which would be to extract sequencing or do something with this DNA. Obviously, if we were trying to collect DNA from a crime scene, this wouldn't be how we would do it. But if we were trying to collect DNA for doing biotechnology, we would want a lot of DNA, and this would be a good way of doing it, because now we've got a lot of starting material, and then we can afford to lose some in the next few steps.